Hello everybody, welcome to my channel. This is Shiva Ravindra. Today I am going to discuss the topic question number 41 to 65 question and answers. So up to 40 questions I already made a video and you can find my YouTube channel those videos, and today I am going to discuss the question number 41 to 65 question and answer. The first 41 to 45 questions related to the concomitant medication related things. Okay, if you done your ADCM data set in your experiences, or you are going to do that thing, so these questions may expect from the interview people, as well as these topics and these scenarios may help you for real time programming purpose also. Okay, so the questions are, what is the prior and concomitant medication, and what are the variables you are derived in the ADAM, and what are the derived variables present in the ADCM, and what is the general imputation rule for ADCM related dates, and what is ATC, and what is the variables, and what is the classification of this ATC. They may ask you this type of question. And whenever you're handling your SGTM data set of CM, if you got an CM class equal to multiple, where you can find that multiple information in your data sets. These questions may roughly, I'm going to explain you. So what is the concomitant medication? As per the definition, the concomitant medication is, here, concomitant medication nothing but other than our study drug if any drug was given during the clinical trial. Okay, so today the clinical trial was started, and after 1 week the clinical trial was ended, for example. So during this one we are even giving some study medication. Apart from that study medication, if any medication was given to the patient to cure the adverse event or any other purpose, that medication we can call it as concomitant medication. Okay, that is a basic definition. So other than the definition, so this SAP mentioned some other definitions also, so I can consider that also. As per my SAP, so they mentioned some other definition, like so if the both start date and stop date of a particular medication is missing, so they can consider as that is also a concomitant medication. Means CM STDTC and CM NDTC both are not collected, or both dates are missing, so that is also they consider as concomitant medication. If the start date of medication is missing and the stop date of the medication falls on or after the first dose of the date, that medication will be considered as a concomitant medication. Means the CMSTDTC is missing and CMNDTC is present, that NDTC is greater than our treatment start date. Means CM end date is greater than our treatment start date. After giving the treatment, if the CM ENDTC was there, so they are considering that event also as a concomitant medication. If start date of medication is missing and stop date of medication is prior to the first dose of day, that medication is considered as not concomitant. Okay, so means if today is the treatment started before, for example, today is 6th September, right? So 6th September is the treatment started. If the event was happening 5th September, so we are not considering that as a concomitant medication. It is considered as prior concomitant. Okay, so it is occurring prior to the treatment started, right? So prior concomitant. So whenever we have a medication occurring during the clinical trial, that is a concomitant medication. Before it happened means any drug was given to the subject, that is called as pre-concomitant medication. If the start date is medication is prior to the first dose and the stop date is after the medication, so the medication also considered as a concomitant. Okay, so medication start date is prior, okay, and the stop date is after. Means yesterday the 5th September is a medication was given, but the stop date, the continuation of the concomitant medication after the treatment started. So that time also we can consider that event as a concomitant. This is as per our general definition along with our SAP definition. Okay. So what are the variables? So variables, you can see here there is a two highlighted one, right? So I'll give you one color. So here on treatment record flag, pre-treatment flag. Pre-treatment flag nothing but, so whenever the medication was happening before taking the treatment, that type of records we are giving as a pre-treatment flag. Whenever the event was occurring during the clinical trial, treatment started and treatment ended, that record we are giving as a on treatment record flag. These are the important variables. I'll tell you how to derive this one also. So what is the derived variables? I'll show you. So you can see this yellow colored one along with the SDTM variables, along ADSL variable. These are the yellow colored one was we are going to derive in the ADC. ASTDT, ASTDY, ASTDTF, analysis end date, analysis end relative day, analysis end date imputation flag, ATC related flags. Okay. So what is the ATC? So ATC nothing but as per this one, there is a some coding terminology also applied to this concomitant medication. So the coding terminology here they are using ATC, anatomic therapeutic chemical classification. Under this they have some dictionaries. So based upon the dictionaries, they will code this type of ATC classes. The based by using CMD code, they will search the CMD code and they will give a specific name as per the ATC classification. Our tables and these things also we are going to do by ATC4 classification by ATC2. How we are doing the AE tables, like AESOC by AED code, how we are doing the same way. The CM medication related coded terms also they are going to do by ATC4 and ATC2. As per this one, they are classified ATC1, 2, 3, 4 like that. So here, where what is the version of this one we used here? So WHODD version 2017 by 12. So based upon which version you are using to generate this type of coding terminology. So you can find it in the listing mark shell footnotes as well as in the SAP also they will mention. Otherwise, you can ask the SDTM people or CDM people to get this one because this is the most important. Interview people may definitely ask you which version you are using for AE decode and AE terminology related concomitant medication and MS terminology etc. version. So here WHODD version 2017 by 12 was used our study. So if it is a CM class is multiple, where we get this information. So for example, here you can see this is a CM SDTM dataset. If CM class is in a multiple, if they mentioned the all the information related to get in a supplementary CM dataset. How we can find whether this is a multiple to related this one? Here you can see the subject number and sequence. Here they mentioned subject number and sequence number. ID where you can see the all the sequence number, and in that they mention all the CM class related and the coding related words. This multiple means there is a lot of concomitant medications associated with this record. So all the class as well as the coding terminology mentioned. And whenever you are doing the ADCM, we can transfer this and back merge to our main data set. So we'll look into the programming so that you can get a clear about all these 5 questions. Okay. First, what we did just running the libraries as well as just keeping the variables from the ADSL. So ADSL, that all the companies they mentioned some specific keeping variables, not to include all the variables. So that is not a particular rule, but you can keep all the variables also due to the space and storage of the server. So for reducing the server and burden of the server, so what the client suggested to use some specific and keep some variables from the ADSL to keep all the other data sets. So as per this suggestion, we are keeping the flags as well as some treatment related variables into the ADSL, keeping only and merge to our main ADCM CM data set. CM also we have these ASTDT and ANDT derivations are there. So those are nothing but just we have to impute that one. So whenever we have a CM partial date, so those variables we are going to impute by imputation rule. If the imputation rule was not mentioned in any SAP, you can confirm first. If they want to use a general rule, I know here I am writing the general imputation rule. General imputation rule nothing but if the date is missing, so simply giving the first date. If the month is missing, if it is a start, January 1st. If it is the end, December 31st. Like that, the general rule I am showing here. If any specific rule is mentioned in my previous video, I'll explain the AEST, DTC and related imputation, right? So you can follow in the same procedure. Here they mentioned the general rule so that if it is a day is missing nothing but. So here the year part is present and month part is present, like 2020 and for example 9th month, the date is missing, so automatically the length is 7. In that cases you will give the 1, means first day of the month. If it is only the year is present, that time January 1st you have to display and create the ASTDTC variable. Okay, in that time you can mention the flag as M because we are replacing the month as well as date. Here we are replacing only the date, so that's why we are putting as a flag is D. So whenever the end date having only the year part and month part, so you can search. If it is a February, you can give as a 28. If it is in a first month, third month, fifth month, you can give a 31st. So if it is a other month, so you can give as a 30. Okay. So that time you have to give the flag D. We are replacing the day part. If it is a month and day is missing, automatically you have to give the December 31st in case of CMENDTC handling AENDTC handling variable. If it is a complete date is present, directly we can convert them into the numeric format and create the ASTDT. And if it is the CMNDTC is a complete date, so we can give the AENDTC and give the sequence number. Okay, this is a simple general scenarios. And wherever we get the ATC classification related variable, all the variables as per the discussion they send into the supplementary data set. So we have to pick from there. So here, so you can see the supplementary CM has this variable. So ID var val, you can see this text1, text2, text3, text4, right? So the QC label is showing that ATC 1, 2, 3, 4 like that. So we have to pick the text3 and text4 and text1 and text2. So text1 related variables are ATC1, text2, ATC2, text3, ATC3, text4, ATC4. Along with this, they are asking line therapy related variables from the supplementary domain as per the specification file. Here you can see line therapy. So line therapy variable also they are writing that, so you can pick up the variables from the supplementary. So I am picking from there and creating the data sets. All these data sets are going to merge here. So before that one, just we are deriving the our main variables like whether the concomitant medication is ongoing, whether the concomitant medication was happening before the treatment. We have to figure it out because all the concomitant medication are coming to the on treatment flag. All the prior concomitant medication comes under this one. So if your listings are table showing separate, that time you have to filter this flag. Okay, prior they want to see the prior concomitant medication, so you have to filter this flag. If they want to see the prior and concomitant medication, both have to select. If they want to see the specific concomitant medication, so you have to select this variable. Okay, so how to derive this one? So simple. So if the AEST for example, AENDTC, so for example, 10th September 2020 is the end date of the treatment, and so treatment start date is, for example, today 6th September. So this AENDT means the concomitant medication end date was 10th September. So if it is a greater than our treatment start date, then and CMNF are equal to whether it is ongoing or unknown, then we can decide it is a ongoing. Okay. So our concomitant medication ended. We created this AENDT variable just now, right? So the after imputation we created this AENDT. So this AENDT is greater than our treatment start date. So that as well as otherwise the CMENRF equal to ongoing or unknown. So we can give as it is still ongoing, means that concomitant was happening during the treatment phase only. So whenever we have a treatment start date, our end date is less than. So for example, it is on 5th September only the concomitant medication was ended. So it is less than our treatment start date, right? So we can give as it is a pre-treatment, means that concomitant medication was started as well as ended before your treatment started. So that time you can give as a pre-treatment flag equal to Y. So in this way, we can differentiate the concomitant medication in a two different ways. After that one, you can merge and you can give the labels as per the specification file and keep the variables which are mentioned in the specification file. And finally, you can send your final data set to the Adam library with the label. So here label also important. So you have to give the label as per the standards. So this is the way ADCM, the overview of ADCM. So most of the ADCM, 90% of the ADCM like this only. So if anything added for new and that is for your study analysis, then only we can add that variable and derive the truths. Okay. So here you can see all the variables are all the next question is all five questions related to the shift table. So generally programmers think that the shift table is very hard, but that is not a hard table. So understanding of the shift table is a major logic for all the programmers. So here I am, we are expecting the shift table questions from like this. So what is the how do you what do you understanding about the shift table and how to create the small n and capital N for big N for shift tables, and if the category is missing, how to display that missing into your table, as well as what are the id variables used in the product transport, and what are the variables you consider to start up your shift table. These questions may expect from the shift table related questions. So here, first of all, are very important in the clinical studies. A shift table is a table that display the number of subjects who are low, normal, or high at baseline and at the shift at post baseline. Write code for the shift table is an easier task if the ADAM dataset used rather than the raw dataset. If everything handled in the ADAM dataset, so this is not much a complex table. So better to ask your ADAM programmer if the variables are not present in the ADLB, because the ADAM is purpose to simplifying the process of the tables listings, right? So we have to pick the variables instead of deriving everything in the tables level. Okay, so what are the variables, major variables to consider? I'll tell you. So you can see the shift table. So as per definition, if you see what is the reference range at the baseline and how many subjects were shifted from here to at the end of the treatment. For example, 100 subjects are there. So their hematology report, out of 100 are normal at baseline, and out of 90 subjects were high at the end of the study. So they are shifted from 90 subjects are shifted from the baseline normal to end of the treatment high ranges. So we can cross comparing with the ranges, how many subjects were shifted from the one positive range to negative range and negative range to positive range. So what do we observe this one? Shift table is the observation of whether the subjects were safe side or not, because every study has a primary secondary objective. So sometimes primary objective is the safety, sometimes efficacy is the primary objective, and the safety concern is the secondary objective. Here the safety concern part, the shift table was generated. So most of the subjects are shifted from the wrong direction for normal to abnormal, normal to high, normal to low. So we can conclude that our subjects are affected by our drug, and their health or laboratory parameters were got changed. So if it is in a positive sign, for example, 100 out of 100 subjects, 90 subjects RBC counts are high. So even they are shifted to and at that treatment if the 90 subjects are in a normal state. So what they mean, that is a positive sign. So it is a safety concern, as well as it showing that pictorial representation, like once see at glance only you can identify how many subjects are shifted from one shift to another shift, and we can conclude that whether really the laboratory parameter will affect due to our treatment or not. We can conclude this one. So here if it is in a missing value, for example, so how to display, for example, no normal range is there. If it is a missing, what we did is you can simply display the missing row here, as well as here also you have to add the missing row. This question will expect from the interview, because the some programmers may don't know how to display this one. So if it is missing also, sometimes you have to display missing if the statistician requires this only. Otherwise we can simply what are the reference ranges are there in the laboratory ADAM dataset, we can use this one. So what are the actual variables to pick up this one? The main and important variables in the ADLB dataset are, so here you can see, so these two, so BNRND and ANRND, these two variables are most important. This is nothing but at the time of baseline what the value of the particular subject, particular parameter having, and this is the what the post baseline, as well as all the values related to the lab reference ranges are containing this variable. So analysis reference range indicator, and this is the baseline reference range indicator. So we can cross comparing this one to get the number of subjects from shifted from the baseline to the post baseline. So these two variables are most important. So we can ask the ADAM person to include these two variables to create the data set. So we can get an easy way to difference. Here you can see writing code a shift table and easier task if ADAM data sets are used. rather than if the raw data set directly providing us to create the shift table. So you have to derive these two variable and you can use the variable to get a shift table. Okay. So and begin and we'll see the in the programming. So what is the put in ID statement? I will see in the here. Okay. So here you can see the first thing is just we have to pick the variables from the ADSL and adding the treatment also, because they are asking the percentage based upon the overall count. So we are counting the overall subjects here and creating the macro variable. So there is a macro variable here. I am creating the proc scale. So you can create by your different method. You know how to create the macro variable in different way, right? So percentage let as well as SQL methods, as calls input methods. So many methods are there. Here I am using SQL method as well as method to you can also create the macro variable by using the calls input x also. So after that one, so we have to filter the data set like variables. They are asking here, our table is asking only chemistry as well as hematology. So here I am picking only the chemistry and hematology. I am not considering any unscheduled visit. As per the cell, they are not asking any unscheduled visit. They are asking only scheduled visits, and it is comes under safety population. Here you can see they are mentioning safety population. So that's why I'm filtering only safety population subject. And the two major important variable BNRIND and ANRIND. After that one, so they are asking last treatment date. Okay, so that's why we are picking the only last param in variables and cross comparing with the BNRND and ANRND. So we'll get the counts. After getting the counts, we have to do the percentage calculation. How the percentage calculation? Small n divided by capital N into 100 will give you the percentage calculation. Here you can see small n divided by this is the big N, and here the 100% up to 1 decimal. So here already we created the macro variable for that count, right? So here the macro variable is the total number of subjects at the end of the study. So as well as this is the count we are getting, small n divided by capital into 100. If it is both are equal, means small n divided by the capital means, so automatically we will get the 100%. So nowhere we have to display the 100.0, everywhere we have to display the 100. If it is a missing value, so we can give the 0 value. Okay, after that one transpose. In the transpose also we are seeing the baseline versus post baseline relations. Okay, that's why they are asking which ID statement you are using. So based upon the cell. Okay, so here our cell showing treatment end, they want to see the low, normal, missing ranges in a column wise. So that's why ID is the ANRD. If they want to put post baseline versus baseline, so that time the baseline variable BNRND you have to mention in the IV variable. Which variable they want to see in the column wise, so that variable has to mention in the ID statement. After that one, you can give the missing values as into the 0 values. So you can give the page number every 17 lines. I am giving a page number so that page truncation won't happen by using this code. After that one, simply write the proc report where it has to go give the ODS and titles and footnotes you can mention. After that one, just give the variables which you have to mention, which you have to display in this one. So define each and every variable. So you already know the how to define everything, right? Because that is the basic concept. This is advanced level of thing. So if you know the proc report section, then only you can use the proc report section to develop the table, right? So you can define everything, give the cell width and etc. as per the cell adjustment. After that one, before above line and below line, this is not in a general version. So that's why I'm giving the before and after line. After that one, close this one. Okay, so close this one. If anything was missing, how to handle? Just hard coding this data set. So for example, if any subject is missing ANRND or BNRND, so you can display, you can assign this one as a missing. Okay, if ANRND equal to missing, so ANRND equal to just replace with the missing, so that this missing category will automatically appear in the output. Okay, no need to apply anything. Okay, so it's the same code with the missing value. Okay, so this is the thing. So first question is how to display the missing ranges. So if ANRND is missing, so you can apply the ANRND equal to missing. So the missing category, the baseline as well as post baseline we have to represent. So what do you understand? So we'll get at a glance how many subjects are shifted from the normal to normal, normal to high, low to high, etc. So in this case we can figure it out the safety of the subject. How to create the begin and other tables? So by using the macro variable creation, I use the proc scale into class method, sometimes calls input x method, sometimes percentage method also used to count the begin and that is used for the percentage calculation. So what are the variables considered in ADLB? ANR and BNR and both are the strong main variables. BNR and containing baseline results related and ANR and all post baseline as well as baseline results. Okay, what you put in ID statement if the cell was showing the baseline and the column the ANR and D variables are a column wise, so we can use the ANR and D in the ID statement. So this is the shift table. Okay. Next question related to adverse event. There are 10 questions. So the 10 questions are nothing but handling of descending order of frequency. For example, if you see here, they mentioned the program note like system argon class is displayed in a descending order of frequency for total then alphabetically. Preferred term is displayed in the descending order of frequency for total then alphabetically within the system argon. What it is means? So we have to display the system argon class and preferred term in a alphabetical order wise. So generally, but here they are asking not the alphabetical, they have to display in the descending order of frequency. After that one you go for the alphabetical. So how to handle this programmatically? So the next question is, so how to handle the missing codator if the AE decoder or AE SOCR any bodies is missing, how to handle the programmatically? So how to categorize the AE toxicity grade and how to give this relatedness. For example, here relatedness means relatedness tables are there, so possible or probable or definitely. So in the SGTA we won't find this information related variable. You can find the variable, but exactly this information was not getting. How to handle in that cases? Okay, so overview of AE table, how the overview, so what is the difference between other AE tables and overview AE table? So what is the general adverse event? What is the difference between the serious adverse event and severe? And what is the n, small n, capital in the adverse event? So what is the Adam variables and difference between the SGTM variables? Okay, so which meta version you use for your A table and where you get that information? These are the 10 most common questions interview people will ask you. So how to answer this one? We'll see one by one. So handling of descending order of frequency, we'll see later. So what is the adverse event? So as per the definition, so adverse event nothing but any unintended untoward reactions happening during the clinical, any event untoward unintended reaction happening during the clinical trial, that was called it as adverse event. So adverse events are common to all clinical trial, but some adverse events are serious, some adverse events are serious, some adverse event leads to death also. Okay, so adverse events are the major concern of the safety. So what is the difference between the serious adverse event and severe adverse event? Here you can see the serious adverse event. So serious adverse event SCA is an untoward medical occurrence. So what is the criteria if the adverse event has a serious? So that adverse event results in a death, is life-threatening, required inpatient hospitalization or prolonging of existing hospitalization, result in a persistent or significant disability, incapability, is in a conventional abnormality, birth defect, is of a medical important. So these criteria, if the adverse event having, so we can call it a serious adverse. What is severe? So we have a based upon an intensity, so we can consider as it a mild, moderate, severe. For example, if a headache was happening as an adverse event, so they have a severity like mild, moderate, severe. So all the severe adverse events are not going to serious. If we are called it as a serious means the below criteria, any one of the criteria met, then only we can call it as a serious adverse event. Okay. So the next question is, here you can see small n. Small n is the number of subjects and number of adverse event. Capital N is the total number of subjects. E is the, for example, E nothing but here you can see the E. So here E represent the number of adverse event. Here you can see first 4 is the subject counts. So infestation and infections and infestation, 4 subjects got. This 4 subject 5 times get the adverse event. Okay. So this is a 4 subject getting this infestation. So total number times, how many times of this adverse event represent the number of events, number of events. So here unique subject count, here number of events means 4 subject, 5 times, 5 adverse events. Okay. So this is the E, small n is the 4, this E is the this one, and total number capital N is nothing but just now we discussed they are getting from the ADSL. This divided by 4 divided by that capital N count give you the 33%, 3 like percentage. So small n is the, so here small n represent the number of subject each level of summarization. Percentages based upon the subject of the safety population means capital N count we get from the safety population of ADSL. The small n divided by capital N into 100 will give the percentage. This E, the bracket E represent the number of adverse event. Okay, number of adverse event. This is the most important. Definitely the interview people may ask you if you've done your adverse event tables. Okay. So here one more question. So we have an ARL variable present in the your SGTM dataset. They are having the values probable, possible, unlike, unrelated like that. But here what happened, the table showing possible, probable and definitely. Okay, so that time you have to see this here whether it is a not related or related. How can we figure it out? So not related, unlikely, unrelated, so all these categories. So not related, unlikely, unrelated comes under not related. So if it is a possible, probable, definitely is a related. So that you can see here they mentioned by relationship. So whether it is a summary of immune response relation, system class, preferred term. Okay, so here possible, probable. So whether it is a not related or related, how can we know that whether it is a not related and related? So this criteria. So related categories are possible, probable and definitely. Not related categories are not related, unlikely, unrelated. So if you are not creating this one, so if they are asking only the subject related to the adverse event, so you don't know. So ARL is there, but you don't know which one is related and which one is not related. So possible, probable, definitely comes under related. Not related, unlikely, unrelated comes under not related category. Okay. So next, overview of the table. So every I adverse event tables are, system architect and preferred term. But overview, we can see how many subjects have adverse event, how many subjects have severe, how many subjects are serious, how many are drug related. Here you can see they mentioned leading to premature discontinuation, serious adverse event. Here you can see the line drug related serious adverse event. So if you are not done this one, so if you are not done this one, not related or related, so you can't do this line drug related, whether it is a drug related or not. So you have to find the subject count. If you write the code, so if you are categorize the code, or if you are develop this rel gray gr1 variable. Okay, so you can give any name, but here as per the standard I giving as a rel group, so not related and related. Okay, so how to handling the descending frequency, I'll show you. So here first of all just keeping the variables from the ADSL and all category, and then creating the macro variable for that one. So next you can see number of subjects with adverse event EAE. Just count the distinct counts. So group by treatment. So SOC counts and preferred terms by counting the SOC counts, group by treatments and bodies is count the unique subjects. Okay. So if you are calculating the preferred term count, that time along with the bodies is you have to mention the AED code also. This is the most important. So after getting the counts, just set all the data sets and count the percentages. So percentage how? So if it is a treatment one, the treatment one macro variable small and divided by that capital N into 100. So if it is a treatment two, the treatment two macro variable 3, 4. We have a 5 treatment, so 5 treatments done. After that one, transpose the data set to category, see the treatment related in a column wise. So after that one, if any treatment was missing, just adding the dummy treatment, so that zero values are producing with the dummy treatment. So the major important steps here after all the getting the count. So here I am mentioning if first dot a bodies is I am creating along these percentages. I am picking the first word before this bracket. So I am converting the value into the numeric. So after completing everything, the simple step, the percentages. If for example 10 and percentages like 33.3. So in this I am picking only the first word means number of the adverse event and created one variable. This is SOC and this is the preferred term counts. So after that one, just giving the order like SOC order and body sys order and preferred term order. So what happened? It will produce the SOC order. So instead of this, if you are writing like descending, so if you are writing like descending indices and descending, so highest values come first, after that one alphabetical. So here also they are asking in that way. First descending order of frequency of total, then alphabetically. So automatically, so if you are giving the descending order of this one count related, next alphabetically, next descending, and get the alphabetically. Okay, so this is a major important step. After that one, the same by statement you can use here, like using this one and counting the page numbers, and you can use the same thing. Okay, so that the descending order you will get. So instead of changing all the codes from every time, at the final step itself you can pick the number from the percentages and convert into the numeric version and using the descending function here. And the same descending function you can use the by statement and create the page number and line number. So you can use the same page number so that nothing was disturbed. So as it is, you can print the descending order of frequency. Okay, so the output may get like this. So I'll show you. For example, so here you can see the 1 1. So it is not comes under alphabetically. So which one having the more value, so it will come first, and which one having the less value, so it will go later. Okay. So the next one, if it is in a handling of missing code, the simply you have to give the code like this. So here you can simply mention if AE decode equal to missing, then AE decode equal to, so you can give some star star so that uncoded. So if it is a body sys also, you can write the same code so that your listing or your table populated this uncoded wise. So you can inform to the medical coder team this terms was not coded, and check that one whether really this code has a new code was getting into the data. Otherwise, due to some oversight, if they are ignoring that code, so you can find out. Okay. We have a mild, moderate, severe there in the A severity, but common terminology criteria. So they will give a category wise this one. So we have 5 categories: mild, moderate, severe, life-threatening or disable AE, and death-related. So this information was mapped to the SDTF AE tox grade variable. So AE severity variable mapped to mild, moderate, severe. But in the CTC AE toxicity level related thing as done, the drug was more toxic or based upon the drug molecule. If they mentioned in the CRF toxicity are also collected, so the doctors will choose in this one of option whether that adverse event toxicity grade. Based upon the toxicity, we have 5 categories: mild, moderate, severe, life-threatening or disable AE, death-related adverse event. Okay. So the next question is relatedness. We already seen overview. Already seen what is AE, serious, captain, and difference between the SGTM and AE variable. So here you can see the something was different from the SGTM variable to AE. So here AESCB is the SGTM variable, ASCB is the Adam variable. Why it is deriving both the times? If anything was missing in the SGTM AE variable, if they are statisticians suggested to impute that variable in a specific scenarios, that time the SCASCB was updated. Apply imputation rule of missing severity of adverse event specified in the SAP or metadata may change the text some such uppercase AES to mixed case of AES. So this is the SGTM and this is the Adam variable. If we are not missing anything, so we can directly all the values are similar to AESCB. I no need to represent this variable if the rule was not mentioned in the SCP. So AREL is the SGTM variable, AREL is the ADAM variable. ATOX grade is the SGTM variable, ATOX grade is the ADAM variable. Okay. So after that one Medra version used, so you can see the Medra version related thing here. So adverse events are coded using Medra version. This information was, you can find it in the footnotes as well as SAP. If you are not getting this information, you can ask the medical coder team or CDN department. They will produce your Medra version. Otherwise, trial domains also, one of your trial domain also containing this Medra dictionary or all the dictionaries information. This is what the information related to adverse event table. Protocol deviation. Question related to the protocol deviation. What is the deviation of protocol? What is the difference between the protocol deviation and violation? And what are the flags we derived based upon this derivation? What is the major protocol? What is the time necessary report, etc. We'll see. Okay, so here what is the difference between the protocol deviation and protocol violation? So deviation nothing but accidental or unintentional changes non-compliance with the research protocol does not increase the risk of this, decrease the benefit. Okay, so in case of something was not happen, something was happening accidental or unintentional changes. Okay, a reschedule of study visit due to unavailability of patients, failure to collect the samples. For example, 2 minutes the sample is going to take. So due to some cannula blockage or anything must happen to the phlebotomy, so that may be 1 minute delay. So that is the deviations. Okay, so in case of violations, accidental or unintentional changes non-compliance with the IRB approval protocol without prior sponsor or IRB approval. Without approval, if you are doing something, or if you are executing the study without proper the approval, so those are the violations. So here something like not following the inclusion and exclusion criteria. So if they are mentioning some inclusion and exclusion criteria to select the candidate, so if you are not following, that is a violation. Accidental disturbances are incorrect study medication dose. If you are planning to give 10 mg, so if you are giving 20 mg to the subject means that is a major protocol violation. So loss of laptop computer, the confidential matter in 2012, private information about the subject, failure to upload the valued information consent. So those are examples of protocol violation. So this is a deviation, this is the violation. All the information we are getting into the protocol deviation data set. So what is the major protocol deviation? So major protocol violation is a deviation that is impact of the subject safety, substantially alert the risk of subject, may have an effect on the integrity of the study data, or may affect the subject willing to participate in the study. So major protocol always the violations of a deviation. So that is impact of the subject safety. Okay, in case of protocol deviations, so what is the variable we have to produce in the ADSL? Per protocol population flag. So per protocol population flag. So here the per protocol population flag PP population usually define all the patients who completing the study without major protocol deviation. So who subject having don't have any major protocol violation. That subject only included the per protocol PPROTF flag. So this is the ADAM variable which are associated to our this deviation data set. Okay. So what is the timeline to report the major protocol? If there is major protocol deviation happening during the clinical trial, so deviations must be reported to the review board within 15 calendar days, and investigator became aware of protocol deviation using the network of REB protocol deviation reporting form. We can report this one. Okay. For example, if it is a protocol violation required to the IRB within the 10 business days of becoming knowledge of the violation. Okay, a letter signed by the investigator, principal investigator, must be submitted with the contains the following information to IRB: project number, subject number, date of etc. Violations are in a 10 days and the deviations are in a 15 calendar days. So this question may will ask you in interview. So the number of variables DB term in a sub-DM is more than one. How to find out? So I'll show you the program. So this is simple. So just picking the variable from the ADSL and DB dataset, and if any supplementary datasets are there, so we can pick up the DB term from there, and checking the variable. So maximum of this DB terms. Just select the how many maximum. Okay, each and every qnum DB term. So maximum number we just converted and creating the variable dv term number, so that if the dv terms are greater than 1 for record, so we can conclude that there is a warning. This number of variables in the dv term in the sub df is more than 1, because if it is a more than deviation, so we are remerging is getting a problem in the deviation data set. So whether this is a warning, because every subject DV sequence 1 has a unique deviations, right? So if it is a more than, means any duplicates are there, we need to check by counting the major DV terms, and that DV term is greater than 1 or not, we need to check. Okay, so that we will get a proper information, and avoiding the duplicates. After that one, just transposing the data set, like remerging with our main data set and create the ASTDT and ANDTT without if it is mentioned any imputation rule for the DB, so we can apply. Otherwise, simply ASTDT equal to DB and treatment variable assigned, and applying the attributes and finalizing the data set. Okay, so this sub DB data set don't have any duplicates records. We need to check in case of deviation, because every subject, every sequence, if it is in a multiple, so that sequence number don't have any duplicates. If deviations have more for one subject, but within the sequence number we don't expect any duplicate deviations. Okay, so here flag is protocol population flag, and major protocol nothing but, so if it is a major protocol, means it is having a major protocol violation has impacting of the safety, then only we can consider that deviation is a major protocol deviation or violation. Okay, all the major protocols may not be DB violations, but all the violations are they are deviations only. Okay, so timelines to report 10 days and 15 days. Okay, so these are the content for this video.